In the last chapter, we covered the non-specific defenses of the host, and now we're going to move on to specific defenses or acquired immunity. Here's a definition. So the adaptive immune system or your acquired immunity, those are defenses that target a specific pathogen. That means it involves a strategy, and that all revolves around lymphocytes. You can acquire this kind of immunity either by getting sick, so you get infected, you get sick, and then you get well and you develop the antibodies, or by vaccination. And I can't say enough positive about vaccination. It is like getting the final exam 2 weeks ahead of time. You're ready and you're prepared and nothing will go wrong. So vaccination is the way to go. If there is a vaccine available, you should get it. And I'm sure right now everybody would love to have a vaccine against this coronavirus, but we don't have one just yet. So we have to wait. It takes some time to develop this. But vaccination is your way of developing immunity, acquired immunity, adaptive immunity, without having to get sick. What could be better? So when you are developing adaptive immunity or acquired immunity, you have a primary response. And the primary response can be either due to you getting exposed to a pathogen, or you getting exposed to an inactivated or attenuated kind of pathogen, and then you develop, so that would be in a vaccination, then you develop your immune response. A secondary response is much faster because you already learned how to respond to this pathogen, and so it's faster, more effective, because you have this immune memory. You saw this pathogen already, you made a strategy already, you know how to deal with this pathogen. Your adaptive immune system revolves around one type of cell, and that will be the lymphocyte. Now, lymphocytes differentiate mostly into B cells and the T cells, and therefore we call it here the dual nature of this adaptive immune system. So starting out with the B cells make antibodies. They got their name from the bursa of Fabricius in birds, this bee right here, therefore called B cells. A lot of people call anything mediated via antibodies, via B cells, humoral immunity. I prefer the name antibody-mediated immunity because it tells you better what it actually does. So B cells, these are lymphocytes that make antibodies. That's the important part. And when they're done making a specific antibody, they keep memory cells around that can be quickly turned into plasma cells that then make active antibodies. Memory cells are sort of dormant cells that can be reactivated at any time. T cells, they are taking care of the cell-mediated immune functions, so therefore called cell-mediated immunity, and these are T lymphocytes. So the two together, B lymphocytes and T lymphocytes, that's the dual nature of your adaptive immune system. T cells engage into sort of cell-to-cell combat, if you will. They have a variety of functions. They need to be activated. They recognize antigenic peptides, and they mature in the thymus if you still have one, if you're young enough to have one. And they have receptors, T cell receptors, and they get in contact with the antigen, and then they typically secrete cytokines to communicate with other and activate other branches of your immune system. They do not make antibodies. That's the job of B cells. So keep in mind, T cells, cell-mediated functions. B cells, they make antibodies. Here's a diagram that shows you the differentiation of T cells and B cells. They all start out with the stem cell, this hematopoietic stem cell. And then if you have a lymphocyte that wants to differentiate into either B cell or T cell, if it's going to be a B cell, you're going to go into the red bone marrow and then differentiate into B cells. If you're going to be a T cell, you're going to go to the thymus and then differentiate into the T cells in the thymus, and that's a mature T cell. Eventually, they all migrate into these lymphoid tissues. And here's your summary. So cellular immunity by our T cells attacks antigens found inside of cells, such as viruses, sometimes fungi or parasites. Humoral immunity via B cells fights invaders outside of cells. And humoral immunity, these are B cells that are making antibodies, and they will fight, then bind to bacteria and toxins using antibodies. Now let's take a look at cytokines. Those are chemical messengers of the immune system. Think of them as kind of like hormones of the immune system used for communication between cells and sort of activating different branches of the immune system. So here's your bunch of definitions. Cytokines, chemical messengers produced in response to a stimulus. Interleukins. Inter means between, leukins, white blood cells. So these are cytokines that are used to communicate between white blood cells. Chemokines. They induce migration of leukocytes, such as diapedesis, calling a bunch of white blood cells into an area of inflammation. Interferons, also cytokines, they interfere with viral infections of host cells. They're used to communicate from one cell to the next that viral attack is imminent, to guard yourself. So interferons, they're mostly associated with viral infections. Tumor necrosis factor, TNF, is involved in the inflammation of autoimmune diseases. Hematopoietic cytokines, hematopoietic cells, these are stem cells that are making all sorts of white blood cells. They control stem cells, develop into red cells, white cells. Overproduction of cytokines can lead to a cytokine storm. And here, as a sideline, you may have heard in the news that COVID-19 produces this massive inflammatory response in some children to the infection, to this virus infection. This cytokine storm can be severe. You may have seen these pictures, images of children that are affected by this. So that means their immune system is reacting so strongly and overproducing all these cytokines that it's producing a cytokine storm that then induces this massive body-wide inflammation. More definitions here for you. An antigen is a substance that causes the production of an antibody. So antigen, antibody. Usually components of invading microorganisms or foreign substances, those will be the trigger. They are those antigens that then trigger the formation of antibodies. Antibodies interact with epitopes or antigenic terminants, whatever was on the antigen that caused the production of the antibody in the first place, and then the antibodies custom made against these epitopes or antigenic determinants that were found on the antigen, a virus or a bacterium, the bacterial surface. A hapten, by contrast, it is kind of an antigen, but on the other hand, it's too small to provoke an immune response. So by itself, it can't really trigger the formation of the antibodies. But a lot of times, if haptens are attached to carrier molecules, now they're big enough to be seen by your immune system, and then they can produce an immune response that leads to formation of antibodies. Here you can see the surface of a bacterial cell, and has these different kind of surface structures that could trigger the formation of an immune response. And here we have an antibody that binds to the triangle structure right here. This is antibody A. You could also make antibodies against these epitopes. All of them could be triggering an immune response, and ultimately lead to the formation of antibodies against the square or this, whatever this round thing is. So here's antibody B binding to this more rounded structure. A hapten, by contrast, by itself, these hapten molecules, they're too small to trigger an immune response. They need to be somehow attached to a carrier molecule to produce an immune response, and then you will get antibodies against this surface structure. By itself, you wouldn't get the formation of antibodies, but attached to some sort of a carrier molecule, then your immune system will make antibodies against this surface structure. Let's take a look at antibodies. Those are globular proteins called immunoglobulin, so that's abbreviated IG. The valence is the number of antigen binding sites on an antibody. Bivalent antibodies have 2 binding sites and so forth, so you can imagine. But the most important term here is immunoglobulin, IG. The most common type of antibody is an IgG. So that's this one right here, the immunoglobulin type G. The IgMs and IgAs are also fairly common. IgMs are basically 5 IgGs together in an arrangement. IgAs are 2 IgGs in an arrangement. But let's take a look at first the basic structure of an IgG, an antibody. You have 4 protein chains that form a letter Y-shaped molecule. You have 2 light chains and 2 heavy chains that are joined together by disulfide links. There is a variable region that is custom made against a specific antibody, and it will bind these epitopes. And then there's a constant region, the stem region, which is identical to a particular class of IgGs. So all human IgGs would be having the same stem region, or all mouse IgGs, they would have the same type of structure in the stem region. Here's the structure of a typical antibody, and in green you're gonna have here they have 2 heavy chains, and in blue the 2 light chains right here. They're held together by these sulfur bridges, as you can see right here, sulfur bridges. And you have up here, only in tip region, you have the variable region. That's the antigen binding site. That's custom made. That's always different. So here you have the antigen, and fits perfectly into the binding pocket here on the tip region of your IgG molecule. That's here, sort of this enlarged region right here, where you have the antigen fitting perfectly into the binding pocket that was created by the antigen binding site on your IgG. Now the stem region right here, that one is identical, at least for a particular species. It can vary if you go to a different species, but for a particular species, within the species, it's the same. Here are the different types of immunoglobulins. By far the most abundant one is the IgG, so the immunoglobulin type G. It's a monomer. It's 80% of the serum antibodies. You find it in blood, lymph, and intestine. They can cross the placenta. They can trigger complement once they bind to their antigen. They can enhance phagocytosis, neutralize toxins and viruses, and they can protect the fetus. IgMs are pentamers made out of 5 monomers, and they held together with a J chain. It almost looks like a snowflake if you take a look at an IgM. So you have 5 IgGs. Try to draw this here so you can see 5 IgGs sort of held together here in the center with a J chain. That's about 6% of your serum antibodies. They remain in blood vessels. They cause clumping or agglutination of cells and viruses. They usually the first response to an infection, and they are short-lived. IgAs, they're monomers in serum and dimers in secretions such as saliva, tears, breast milk. They're about 13% of your serum antibodies, and they prevent microbial attachment to mucous membranes. It's sort of this resident population of IgAs, let's say, in your tear fluid that prevents your eyes from being infected. The other 2 types of immunoglobulins are rare. The IgD here, it's a monomer. It's only 0.02% of serum antibodies. Structure similar to IgGs, and you find it in various tissues. And the function is not well defined, so we can just move on. IgEs are also monomers, even rarer, only 0.002% of serum antibodies. They can be on mast cells, on basophils, and blood, and they're associated with the release of histamines when bound to antigens, and they can cause lysis of parasitic worms. Here's your summary of the types of immunoglobulins. So the IgG is by far the most important one. It's 80% of those immunoglobulins. and it looks like a letter Y. The IgM looks like a little snowflake with its 5 IgG sort of connected with a disulfide bond, and the IgA is also important. That one here you find in secretions such as tear fluids. IgDs and IgEs are not that important for the purpose of this class.